Very good morning to all of you. Today we are going to see a part that is associated with again oxidation, but this time instead of the carbon molecules, it will be oxidation of fatty acid. Basically, we are going to see the beta oxidation of fatty acid. So let us understand what is fatty acids. Beta oxidation may be defined as the oxidation of fatty acids on beta carbon atoms. So there are various different carbon atoms. Basically, the position of the carbon atoms is been defined, and therefore we say that it is the beta carbon atom where the oxidation process of fatty acid would take place, and therefore it is called as beta oxidation. This results in the sequential removal of two carbon fragments, acetyl coenzyme A. Now, there are three stages basically that are associated with the beta oxidation, and in which tissues the beta oxidation would take place. Which tissues do not support beta oxidation, that we'll try to study. So, activation of fatty acids occurring in the cytosol, transportation of fatty acid into the mitochondria, beta oxidation proper in the mitochondrial matrix. So, beta oxidation occurs in the mitochondrial matrix. Fatty acids are oxidized by most of the tissues in the body. Maximum of the tissues are oxidizing the fatty acids in order to gain some kind of energy. But there are certain parts inside the human body which do not oxidize this fatty acids, which is brain, erythrocytes, and adrenal medulla cannot utilize fatty acids for the energy requirement. Now, how the activation would take place? Fatty acids are activated by acetyl coenzyme A by thiokinases. This is a particular kind of enzyme that is associated with the beta oxidation, or acyl coenzyme A synthase. The reaction occurs in two steps and requires ATP, coenzyme A, and Mg2 plus ions. Fatty acid reacts with ATP to form acyl adenylate, which then combines with coenzyme A to produce acyl coenzyme A. Now, based on this reaction, if you see this. Fatty acids with the help of thiokinase, pyrophosphates are being released. Pyrophosphatase is an enzyme. The 1 ATP molecule is being utilized here, and acyl adenylate is being generated. From this acid adenylate, another coenzyme A acid molecule is being released and is added. And this coenzyme A, once is there, the AMP has been removed out, and acyl CoA has been formed. Now, the inner mitochondrial membrane is impermeable to fatty acids. So, inside the mitochondria directly cannot go. So, there should be some carrier system which has to carry these molecules inside. So, what are they? The special carnitine carrier system, that is carnitine shuttle, operates to transport activated fatty acids from cytosol to the mitochondria. See this. Carnitine transport system. From cytosol with the help of acid and the enzyme. Here the mentioning of the enzyme is there. Acyl group of acyl coenzyme A is transferred to carnitine, catalyzed by carnitine acyl transferase, that is CAT. This is CAT1, present on the outer surface of the inner mitochondrial membrane. The acyl carnitine is transported across the membrane to mitochondrial matrix by specific cardiac proteins. Carnitine acyl transferase 2 found on the inner surface of the mitochondria. So CAT2. Acyl coenzyme from the cytosol comes in the matrix, mitochondrial matrix. Now, this beta oxidation occurs in various different stages. So, what are they? Liberating a two carbon unit, acyl coenzyme occurs in sequence of 4 reactions. First reaction is oxidation, hydration, again oxidation, and cleavage. First is oxidation. Acyl coenzyme undergoes dehydrogenation by a FAD-dependent flavoenzyme, acyl coenzyme A dehydrogenase. A double bond is formed between alpha and beta carbon. Hydration, enoyl coenzyme A hydratase brings about the hydration of double bond to form beta hydroxyl coenzyme A. Oxidation, beta hydroxyl acyl coenzyme A dehydrogenase catalyzes the second oxidation and generates NADH. The product formed is beta keto acyl coenzyme A. Cleavage, the final reaction in beta oxidation is the liberation of two carbon fragments, acyl coenzyme A from acyl coenzyme A. This occurs by thiolytic cleavage catalyzed by beta keto acyl coenzyme A thiolase, specific enzyme. New acyl coenzyme A containing two carbon less than the original re-enters the beta oxidation cycle. Process continues till the fatty acid is completely oxidized. See here. Fatty acids with the help of thiokinase from the cytosol it enters as acyl coenzyme A into the mitochondria. So once inside the mitochondria, the acyl coenzyme A with the help of an enzyme, acyl coenzyme A dehydrogenase, get transferred to trans-enoyl coenzyme A. And trans-enoyl coenzyme A goes to beta-hydroxyl coenzyme A. Once this has been done, the beta-hydroxyl coenzyme A undergoes again a transformation with the help of an enzyme, beta-hydroxyl acyl coenzyme A dehydrogenase, to form beta-keto-acyl coenzyme A. Again, thiolase is there, which splits this beta-keto-acyl coenzyme into two parts. One is acyl coenzyme A, another is acetyl coenzyme A. Sorry. This acetyl coenzyme A is now funneled into the TCA cycle, and the acyl coenzyme A is again funneled back to take another carrier molecule of the the fatty acid and bringing it to the inside the site into the mitochondrial matrix. Correct? So that's all for the day. This is the basic part that we have learned regarding the beta oxidation of fatty acids. Next lecture, we will try to study in greater details the important information about beta oxidation of fatty acids.